Hello everyone, welcome back to my lectures. So this particular video is about expression vectors. So let's have an introduction. Molecular cloning and genetic studies require the use of vectors as a host or carrier system to transfer selected genetic components to cells or organisms. The expression vectors act as vehicles for DNA insert and also allow the DNA insert to be expressed efficiently. They are genetically engineered for the introduction of genes into the target cells and also contain regulatory elements like enhancers and promoters for the efficient transcription and translation of genes of interest. The simplest expression constructs are also known as transcription factors because they allow transcription of the cloned foreign gene and not its translation. The vectors which facilitate both transcription and translation of the cloned foreign gene are known as protein expression vectors, which lead to the production of recombinant protein. These vectors are frequently engineered to maintain regulatory sequences that act as promoters or enhancer regions and lead to efficient transcription of the insert gene. And essential features of expression vectors. The gene of interest, which is shortly represented with the help of acronym called GOI, expression, has it with promoter and a gene termination or poly A signal. So prokaryotes expression vectors would have a Shine Delgado sequence at its translation initiation site for the binding of ribosomes, while eukaryotes expression vectors would contain the Cossack sequence. And promoter is a sequence which is recognized by sigma subunit of RNA polymerase, which is required for initiation of transcription of gene of interest. The promoter initiates the transcription and is therefore the point of control for the expression of the cloned gene. The promoters used in expression vector are normally inducible. Promoter synthesis is only initiated when required by the introduction of an inducer such as IPTG. So constitutive protein is constantly expressed in some expression vectors. Low level of constitutive protein synthesis may occur even in expression vectors with tightly controlled promoters. Then terminator, it is a DNA element present at the end of a gene where transcription of gene ends. Terminator is short nucleotide sequence which can base pair with itself to form hairpin loop. And ribosome binding site, it is a short nucleotide sequence recognized by the ribosome as the point at which it should attach to the messenger molecule. The initial codon of the gene is always a few nucleotide downstream of this. Multiple cloning site MCS, purification tags, his tag, after the expression of the gene product to make this purification process easier, a purification tag may be added to the cloned gene. This tag could be histidine tag, other marker peptides, or a fusion partner such as glutathione S transferase or maltose binding protein. Some of these fusion partners may also help to increase the solubility of some expressed proteins. Other fusion proteins, such as green fluorescent protein, may act as a reporter gene for the identification of successful cloned genes, or they may be used to study protein expression in cellular imaging. So here we could see the key features of a typical expression vector. Okay. Then next is about expression systems. In prokaryotes, where there is no nuclear membrane, both transcription and translation occur in the cytoplasm, where in eukaryotes, transcription occurs inside the nucleus and the translation occurs in the cytoplasm. In constitutive expression, housekeeping genes are essential and necessary for sustaining life and are therefore continuously expressed. Glyceraldehyde 3 phosphate dehydrogenase SDHA, succinate dehydrogenase, etc. are human housekeeping genes which are expressed throughout the development. Then induction and repression. The expression levels of some genes fluctuate in response to external signals. Also, under the certain situations, some genes show higher expression levels while others show lower expression levels. The former is called induced expression and the later is called repressed expression. So, types of expression vectors: prokaryotic expression vectors, bacterial expression system, E. coli, the PET vector system. The PET vector system is a powerful and widely used system for expressing recombinant proteins in E. coli. The gene of interest is cloned into the PET vector under the control of the strong bacteriophage T7 transcription and translation regulatory system. Activation of expression is achieved by providing T7 RNA polymerase within the cell. When the system is fully induced, nearly all of the cell's resources are devoted to expressing the gene of interest. PET vector system is a cloning and expression vector system for recombinant protein production in E. coli. The original PET vector system was constructed by Studia and colleagues. So PET system now available for the expression of proteins. Then COL1E1 origin of replication and F1 origin of replication allows single-stranded vector to be produced when co-infected with M13 helper phage. T7 transcription promoter specific for phage T7 RNA polymerase. Target genes are cloned under strong T7 bacteriophage promoter. The expression of the target protein is inducible by providing T7 RNA polymerase in the host cell as an inducing signal. Target gene is initially cloned to the host cell that do not contain T7 RNA polymerase, thus increasing plasmid stability. When stabilized in a non-expression host, the recombinant plasmid is transferred to an expression host having T7 RNA polymerase gene in the genome. LAC1G, LAC repressor protein, and LAC repressor region 3' to the T7 promoter. Multiple cloning site MCS downstream of the T7 promoter. Ampicillin and kanamycin resistance genes as selection marker. Then PET28 and PET32 are the most commonly used PET vectors. Then eukaryotic expression vector, yeast expression systems, Saccharomyces cerevisiae. Then B, viral receptors or insect cell expression system like baculovirus. Then plant expression system, mammalian cell expression system, yeast expression system, Saccharomyces cerevisiae, cloning and expression of a gene using yeast system has several advantages over E. coli system due to presence of eukaryotic post-translational modification machinery. An expression of complex proteins with the proper modification and folding can be achieved by yeast eukaryotic system. These vectors have yeast origin of replication for replication and maintenance in the yeast system and bacterial ori for maintaining inside a bacterial system. Then different types of yeast vectors include YIP, yeast integrative plasmid, can integrate to the host genome by homologous recombination. This generally yields a single copy of recombinant vector DNA integrated to the host genome. Then YRP, yeast replicating plasmid, YRP are used to obtain a high copy number inside the host, up to 100 copy number. YCP, yeast centromere plasmid. YCP possesses a centromeric region in addition to ARS which facilitate the mitotic segregation of the linear plasmid during replication. The copy number of this vector is essentially 1 per cell. So YEP, yeast episomal plasmid, can be maintained in the yeast system as an autonomously replicating episomal plasmid. This vector contains a part of 2Q plasmid, which is essential for autonomous replication of the vector inside the yeast. The other characters of YEP: the plasmid is circular mode. The plasmid size is 7.769 kilobase pair, and it has COIE1 origin for bacterial manipulation. It has bacterial selection marker gene. It has URA3 as a selection marker gene for yeast cell. It has 2 mu plasmid DNA segment with STB chunk, 2000 base pair long, for application in yeast cells. This helps of the plasmid to replicate in the nucleus and produce a copy number of 200 to 200 per cell. It also contains few cloning sites such as SMA, XMA, PPU, and CLA1 and AAT2. An yeast episomal cloning vector with a 2 mu origin of replication and a URA3 marker. YAC vectors, yeast artificial chromosomes, are plasmid shuttle vectors capable of replicating and being selected in common bacterial hosts such as ESC Escherichia coli as well as in the budding yeast Saccharomyces cerevisiae. Circular map of plasmid vector PYAC3. PYAC3 vector was constructed by Burke et al. In 1987. The basic structural features of YAC were developed from the yeast centromere shuttle plasmids YCP series. These are composed of double-stranded circular DNA sequences carrying the beta-lactamase gene BLA and the bacterial PMB1 origin of replication, thus conferring resistance to ampicillin and the ability to replicate in bacteria respectively. They also include yeast ARS1 with its associated CEN4 DNA sequence as well as the URA3 selectable marker. In this basic scaffold plasmid, the yeast HIS3 is cloned, then flanked by a telomere-like DNA sequence derived from the termini of the tetrahymena piriformis macronuclear ribosomal DNA, which allows the formation of functional telomeres in yeast. There are adjacent to two recognition sites for the BAM-H1 restriction enzyme. And viral vectors are highly efficient in transferring target DNA or RNA segment to the host cell with high specificity. Wild type virus is modified by deleting the non-essential genes and incorporating exogenous nucleic acid segments to construct a viral vector. And viral vectors have wide application in gene therapy and targeted drug delivery system. Main advantages of viral vectors is high transfer efficiency and high cell specificity. Although there are certain safety issues associated with viral vectors and careful handling is essential during the experimental procedure. And commonly used viral vectors are adenovirus, retrovirus, lentivirus, adeno-associated virus AAV, Herpes simplex virus, and simian virus. And simian virus 40 in detail. So simian virus 40 SV40 was the first mammalian expression vector whose genome size is 5.2 kilobase pair. It is a DNA virus which can infect human as well as other mammalian cell lines. SA40 may integrate into the host genome, permitting stable transmission of insert DNA to daughter cells. So recombinant SV40 vectors display some unique features. SV40 is a well-known virus and non-replicative vectors are easy to make. SV40 can be produced in high titers, then they can infect both resting and dividing cells. Stable transgene expression can be achieved in a wide range of cell types. And along with that, the major disadvantages of SV40 vector is the low packaging capacity with insert size of less than 5 kb. Then baculovirus vectors. Baculovirus is an enveloped double-stranded DNA virus that belongs to the Baculoviridae family with double-stranded circular supercoiled DNA molecules in a rod-shaped capsid. The baculovirus expression system has been used extensively for the expression of recombinant proteins in insect cells. Two of the isolates used in foreign gene expression are Autographa californica multiple nuclear polyherosis virus and Bombyx mori silkworm nuclear polyhedrosis virus. Then Autographa californica nucleopolyhedrovirus is the most extensively studied virus under this family. Then genome size of these viruses is about 128 to 1000 kilobase pair and in circular mode. So insect cell lines used for the expression of viral genomes are Spodophora fujiparida, 9 cell lines. On infection, viruses multiply within epithelial cells of gut. So viral genes are expressed in temporal fashion such as early, late and very late. So early gene products initiate its DNA replication and also activate the expression of late genes. This results in the multiplication of the genome as well as viruses, which in turn are budded off as enveloped viruses, so which further infect a new set of cells. So expression of very late genes is about 18 to 20 hours after infection, which code for polyhedron proteins. At this stage, the host cell nuclear membrane proliferates and primary viruses are occluded among the polyhedron matrix proteins. Here we could see a picture which depicts the baculoviral vector. The infection of ACMVPV is initiated by replication and transcription of the DNA genome inside the nucleus and the assembly of the nucleocapsids. The nucleocapsids then bud off from the plasma membrane and initiate systemic infection. Although baculoviral vectors can transfect only insect cells, recombinant baculoviral vectors have been constructed containing mammalian cell-specific promoters which can be used to infect mammalian cells as well. So advantages of baculoviral vectors: since insect cells are high eukaryotes, desired post-translational modifications of complex protein can be achieved. They have higher packaging capability of insert, lower biosafety issue, high level of protein expression. There are few drawbacks of baculoviral vectors, such as foreign protein expression using an insect system is more complex and time consuming than a bacterial system. Sometimes protein post-translational processing may be suboptimal to compensate the secretory pathway of the protein. In mammalian expression system, mammalian expression vectors often offer considerable advantages for the expression of mammalian proteins over bacterial expression systems. Then proper folding, post-translation modifications and relevant enzymatic activity. It is of particular use in producing membrane-associated proteins that require chaperones for proper folding and stability, as well as containing numerous post-translational modifications. In cultured mammalian cell lines such as the China's hamster ovary, CHOCOS, so fibroblast-like cell lines derived from monkey kidney tissue, including human cell lines such as HEK and HeLa may used to produce protein. HeLa cells we know. So vectors are transfected into the VEC cells and the DNA may be integrated into the genome by homologous recombination in the case of stable transfection, or the cells may be transiently transfected. Examples of mammalian expression vectors include the adenoviral vectors, the PSV and the PCMV series of plasmid vectors, Accinia and retroviral vectors, as well as baculovirus. The promoters of cytomegalovirus and SV40 are commonly used in mammalian expression vectors to drive gene expression. So non-viral promoters such as the elongation factor EF1 promoter is also known, a constitutive or inducible promoter that is capable of robust transcriptional activity, a transcription terminator that stabilizes the transcription and prevents transcription interference, optimized mRNA processing and translational signals that include the Kazakh sequence, translation termination codon, mRNA cleavage, and polyadenylation signals, as well as mRNA splicing signals for higher levels of expression. So prokaryotic origin of replication and selection marker for vector propagation in bacteria, selection markers for the preparation of stable cell lines and for gene amplification. The inclusion of the SV40 origin of replication facilitate transient gene expression in COS cells. Other genetic elements for specific applications include consequences for gene or protein targeting, signal peptides for protein secretion, fusion moieties and protease cleavage sites, and ribosome or protease recognition sites that facilitate the expression of multiple genes from polycystonic or monocystonic constructs. So mammalian expression vector requires vectors derived from mammalian viruses such as simian viruses, polyomavirus, herpes virus, and papovirus. So in order to construct vector, the requirement is to select an efficient promoter and also the selection marker. Mammalian expression vector pcDNA, one with CMV promoter, human cytomegalovirus promoter. Promoter derived from T7 phage MCS. The MCS is the forward orientation, refers to orientation of 1 ORI replicates by PUC ORI. Bovine growth hormone polyadenylation sequence ORI of replication derived F1 phage, ORI of replication from SV40 neomycin resistant gene, and SV40 polyadenylation sequence origin of replication for bacterial cell and ampicillin resistant gene. Then plant expression system. Many plant expression vectors are based on the TI plasmid of Agarobacterium tumefensis. In these expression vectors, DNA to be inserted into plant is cloned into the tDNA, a stretch of DNA flanked by a 25 base pair direct repeat sequence at either end and which can integrate into the plant genome. The tDNA also contains a selectable marker. The agrobacterium provides a mechanism for transformation integration of into the plant genome, and the promoters for this VAR genes may also be used for the cloned genes. So concerns over the transfer of bacterial or viral genetic material into the plant, however, have led to the development of vectors called intragenic vectors, whereby functional equivalents of plant genome are used so that there is no transfer of genetic material from an alien species into the plant. Examples of plant virus used are the tobacco mosaic virus, potato virus, and cowpea mosaic virus. The protein may be expressed as a fusion to the coat protein of the virus and is displayed on the surface of assembled viral particles, or as an unassisted protein that accumulates within the plant. So, expression in plant using plant vectors is often constitutive and is commonly used to constitute a promoter in plant expression vectors is the cauliflower mosaic virus or CAMV 35S promoter. Then cell-free systems. E. coli cell lysate containing the cellular components required for transcription and translation are used in this in vitro method of protein production. The advantage of such system is that protein may be produced much faster than those produced in vivo, since it does not require time to culture the cells, but it is also more expensive. So vectors used for E. coli expression can be used in this system, although specifically designed vectors for this system are also available. So eukaryotic cell extracts may also be used in other cell-free systems, for example, the wheat germ cell-free expression system. So mammalian cell-free systems have also been produced. At present, gene cloning in mammals is carried out for one of the three reasons: to achieve a gene knockout, which is an important technique used to help determine the function of an unidentified gene, for production of recombinant protein in a mammalian cell culture, and in the related technique of framing, which involves genetic engineering of a farming, which involves genetic engineering of a farm animal so that it synthesizes an important protein such as a pharmacon, pharmaceutical, I'm sorry, pharmaceutical, often in its milk, in gene therapy, in which human cells are engineered in order to treat a disease, and applications of expression vectors. The last part. Laboratory use. Expression vectors in an expression host is now the usual method used in laboratories to produce proteins for research. Most proteins are produced in E. coli, but for glycosylated proteins and those with disulfide bonds, yeast, baculovirus, and mammalian systems may be used. So, production of peptide and protein pharmaceuticals. Most protein pharmaceuticals like hormones, vaccines, antibiotics, antibodies, and enzymes are now produced using expression vectors. This peptide and protein pharmaceuticals may be the first human recombinant protein used for disease management. Insulin was introduced in 1982. Transgenic plants and animals. Expression vectors have been used to introduce specific genes into plants and animals to produce transgenic organisms. In agriculture, it is used to produce transgenic plants. So, expression vectors have been used to produce a vitamin A precursor beta carotene into rice plants. This product is called golden rice. This process has also been used to introduce a gene into plants that produce an insecticide called Bacillus thuringiensis toxin or Bt toxin, which reduces the need for farmers to apply insecticide since it is produced by the modified organism. So, expression vectors are used to extend the ripeness of tomatoes by altering the plant so that it produces less of the chemical that causes the tomatoes to rot. So genetically modified food. Transgenic animals have also been produced to study animal biotechnological processes and human diseases, or used to produce pharmaceuticals and other proteins. They may also be engineered to have advantages or useful traits. Green fluorescent protein is sometimes used as tags, which results in protein that can fluoresce, and this have been exploited commercially to produce the fluorescent glowfish. So, gene therapy, it is a promising treatment for a number of diseases where a normal gene carried by the vector is inserted into the genome or replace an abnormal gene or supplement the expression of particular gene. So viral vectors are genetically used, but either non-viral methods of delivery are being developed.